In this video, we will learn about the seed germination. Now we know that once the seed is sown inside the soil, it will slowly start to germinate and give rise to a small plantlet. But once we have got the plantlet, it will grow very swiftly. But when we see the seed contains an embryo inside it and it remains inactive or dormant. And if there is any favorable conditions, it becomes active and will grow into a small seedling. The favorable condition include the sufficient amount of water, air and also temperature. Now this process by which an inactive embryo in the seed becomes active and starts to grow is termed as seed germination. Thus we can tell that seed germination is the process during which the food reserves present in the seed are used by the embryo and it starts to grow into a seedling. Now if we take the reserve food in the seed, so the seed is dormant, so it has to grow once the dormant period is over. So once the development is done, the seed growth stops. So once the seed growth stops, it will begin to lose its water content. Now the seed coat becomes impermeable to both air and moisture. At this stage, we can see that all the physiological activities will slow down and very little food is used by the embryo. Even respiration rate is very slow and it will be anaerobic in nature. In this dry condition, the seeds are said to be dormant and the stage is known as dormancy. Now, a freshly formed seed normally does not germinate. So, if I grow a plant and pluck the seeds immediately and put it in soil, it will never germinate because it is not having favorable condition. One. Second, it is not mature enough. Now, even if you give a favorable condition, it will not grow because of the maturity period. So a seed must pass through a period of dormancy during which it undergoes certain physiological changes. Only then it will be able to germinate. Now based on how the germination process proceeds, we have two types of germination. One is epigeal germination, the second is known as hypogeal germination. So this is based on whether the cotyledon remains below the soil after germination or not. Second is which part of the seed germinates faster, that is it the epicotyle which grows faster or hypocotyle which grows faster. The first one is known as the epigeal germination. Now epi means above. So in this type, the cotyledons will come above the surface of the soil once the germination has started and light will be received immediately and due to the rapid growth and elongation of the hypocotyle. Now the difference is for epigeal germination, it is the hypocotyle which will grow faster. So this below part will grow faster and it will push the plumule and the cotyledons above. So cotyledons will turn green and make food for little period of time and that food is utilized by the growing stem. Now they finally dry up, that is you can see here the cotyledons have finally been dried up and they fall off and the seedling will become more and more independent. Examples include bean, gourd, castor, cotton, tamarind, etc. Now the second type is opposite, that is hypogeal. That means here the epicotyle will germinate faster or grows faster. So in this germination, seed will remain inside the soil forever. This is due to the epicotyle elongates faster than the hypocotyle. So the cotyledons need not appear on the soil. And examples include gram, pea, maize, rice, etc. I hope this topic is clear to you. Thank you.